In this video, we are going to continue our discussion of catabolism, which is the breakdown of large molecules such as lipids, carbohydrates, and proteins into ATP. So far, we have talked about the structure of ATP and how it can be coupled to reactions. And then we've also talked about our reduced coenzymes, which are going to be NADH and FADH2. So now with all of that knowledge in mind, we are finally ready to dive deep into stage 3, which is going to be the citric acid cycle. A note here that the citric acid cycle goes by 3 different names. The other 2 are going to be the TCA cycle, which stands for tricarboxylic acid cycle, and also the Krebs cycle. You will often hear me refer to this cycle as the TCA cycle. All right, so first let's look at an overview of this cycle, and then we will dive a bit more deeply into what is happening in each one of these reactions. Okay, so the numbers 1, 2, 3, and 4, this is referring to reaction number 1, 2, and so on in the cycle until you reach reaction number 8. But do note that each one of these reactions is carried out by an enzyme. So there's an enzyme that participates in reaction 1, and then an enzyme that participates in reaction 2, and so on. All right, so the TCA cycle is going to be a cyclic pathway. So what this means is that even though we're starting and we've designated this reaction as 1, we're going to go all the way around and regenerate the starting material so that then we can proceed through another turn of our cycle. So how this works is that our cycle begins with acetyl-CoA entering this cycle. So here we are showing that there are 2 carbons coming in. These 2 carbons are coming from your acetyl-CoA. Recall that the structure of your acetyl-CoA is 2 carbons with a carbonyl here, and then the CoA enzyme, or the CoA molecule is bound to those 2 carbons. Okay, so this is what your acetyl-CoA molecule looks like, and this is carbon number 1, and this is carbon number 2. So those are the 2 carbons that are being shown here in the schematic. So what's going to happen is these 2 carbons are going to join these other 4 carbons that are part of the cycle, and that is going to be converted to a 6-carbon compound, because 4 from here and the 2 from there will give you 6 carbons. Right? So remember that these 2 carbons that are in your acetyl-CoA molecule, these are coming from your building blocks. So what this means is that if you consume carbohydrate, for example, those 2 carbons would come from your glucose molecule. Or if you consume lipids, specifically the triglycerides, 2 of those carbons are going to come from your fatty acids. Right? So that is what we mean by the acetyl-CoA feeds into this cycle. It's going to be these 2 carbons that are coming from the building blocks of your main biological macromolecules. All right, so what are the other points here? So it's important to note here that the cycle ends once you reach this 4-carbon substrate. Okay, so you're going to start by adding these 2 from acetyl-CoA. That gives you 6 carbons. And then as you proceed through the cycle, you reduce those carbons down until you reach this 4 again. And that will be the end of your cycle. And then you would need to feed in another acetyl-CoA molecule in order to do another turn of the cycle. So every time you go once through the cycle, we call that a turn. In order to do another turn, you would need another acetyl-CoA molecule to be fed in, so you can generate the 6-carbon species and then continue. Okay, so here what this schematic is showing you are the carbons. It's tracking the carbons at each step. So if you notice here in step number 3, we are going from a compound that has 6 carbons to one that has 5 carbons. And how that happens is we release the CO2 in this step, bringing our compound down to now 5 carbons. And then in the very next step, we're going to release another carbon dioxide, which is going to bring our compound down now to 4 carbons. In this process, we are also going to be producing our reduced coenzymes, which are the point of this cycle, because these are the ones that are going to feed into the electron transport chain. So in this step here, we see that we produce NADH here as we simultaneously produce the carbon dioxide, another NADH here, right? This carbon dioxide is the carbon dioxide that we release when we breathe out. This is the waste that we release as carbon dioxide. All right, so if we continue through, we're going to see some other important products. This is also going to be a conversion here in step number 5, where we are going to produce GTP. GTP is analogous to ATP. because this TP part is still the triphosphate part. So you can hydrolyze GTP just like we hydrolyzed ATP. We can hydrolyze GTP to be converted to GDP, so a diphosphate, right? And that is going to release energy just like ATP hydrolysis did. We can also synthesize GTP by going in the reverse direction, which is going to require energy. So what is happening here in step 5 is that we are carrying out a reaction where GTP is able to be produced. In the next step, we're going to generate another important coenzyme, which is going to be this FADH2 that will also feed into the electron transport chain. And then over here in the very last stage 8, we are going to produce another NADH molecule. Okay. All right, so this is the zoomed out version of what is happening in the citric acid cycle, with a focus on the main reduced coenzymes that are being generated. We're also generating GTP, and we're also generating these carbon dioxide molecules. So this is going to be one of the main focuses in our analysis today. Right? Now, these are all of the details. I have placed that map that we saw in the previous slide down here just to keep us oriented in what we are looking at. Okay? But now at each stage, we have put in the actual compounds that are being converted from one species to another. Right? So let's start with equation number 1 or step number 1. All right, so here I am showing you acetyl-CoA. It's acetyl-CoA because I recognize that this region here is your CoA enzyme, and it is bound to this acetyl group. Again, an acetyl group is just these 2 carbons with that carbonyl compound there. All right, so this is my acetyl-CoA molecule. I'm going to go ahead and write that in. So this is acetyl-CoA. And these are the 2 carbons. These 2 carbons in blue are the ones that are being fed in to create this citrate. So this acetyl-CoA is reacting with oxaloacetate, which is this molecule here, with also some water. These 3 species are going into your enzyme, and then your enzyme is helping convert this acetyl-CoA and oxaloacetate into one compound, which is called citrate. Notice that during this process, we release coenzyme A. So what is happening here is that this bond here is being cleaved. And because that is a high energy bond, when that bond is broken, we release energy. And it is this release in energy that is helping oxaloacetate and the acetyl group come together into citrate. All right, so let's continue looking at what is going on in the next steps. So you're going to see here in this region shaded in gray, these are the regions that are changing from one step to the other. So let's look at what's happening in the next step. So in this situation, we have our 2 carbons in blue shown here that are coming from your acetyl-CoA. And these green carbons here were existing in your oxaloacetate. Okay, so we're going to be tracking these carbons to kind of get an idea as to what is going on. Okay, so in this step, you're going to notice that the difference here, highlighted in this gray region, is going to be the location of this OH and one of the H's here. They have been swapped. Okay, so that is the change that is happening here in step number 2. So now we look at step number 3, and what we see is that we have an NAD plus coming in, but we also have CO2 being released. So this CO2 corresponds to this group here that is now changing, that is going to leave. And in that process, NAD plus is going to be reduced to NADH, right? So that is this step here. Okay, so if we continue to look at our molecules, we see that now this CO2 has left here. So now we are at this compound. Okay, so I still have this CO2 group here, which is shown down here. Okay, so now this is going to be the group that is going to leave in the next step of the citric acid cycle. So you can see here that this CO2 is now leaving, which corresponded to this CO2 group here. And in that process, what is coming in is another acetyl-CoA. So if you look closely at your product in this reaction here, what you'll see is that this CO2 group down here has been substituted with your coenzyme A. So in this reaction, we release CO2, but in that process, we are able to attach this coenzyme A to my molecule. In addition, we were able to produce this reduced coenzyme by NAD plus coming in and being reduced to NADH. Okay, so now we're at this point here where we have lost 2 carbon dioxide molecules. So we are down to 4 carbons now. Okay, so we are at the point where we have 4 carbons because we have lost 2 of them as carbon dioxide. Up here, we started with 6 carbons. All right, so let's continue. So now let's look at what is happening in stage number 5. So in step number 5, we are going from this compound, which contains this high energy bond, which is the coenzyme A bond here, this thioester bond. When we track what is happening between this step and this one, what we see is that we are releasing this coenzyme A, which again means that we have broken this high energy bond here. So when we break that high energy bond, there is energy being released. Well, what is that energy being used for? Well, you can see here that GDP is coming in and it's being phosphorylated to GTP. So what this means is that a reaction, such as the phosphorylation of GDP, that would normally require energy, by hydrolyzing this high energy bond with coenzyme A, that thioester bond is going to provide the energy that is needed to phosphorylate GTP. So again, GTP, you can think of this as analogous to ATP. It eventually gets converted to that. All right, so in this step, in step number 5, we were able to achieve the production of GTP. All right, so now let's continue. So now we're here at succinate. Now, if we look closely at what is going on between succinate and the product fumarate, we look here in this shaded gray region, we see that 2 hydrogens have been removed in order to create an alkene in this step. So here we have created an alkene. And we were able to do that because we were able to oxidize succinate into fumarate by using our oxidizing agent FAD. So FAD is converted to FADH2 because it took these hydrogens from succinate. And now we are at fumarate, which has that double bond. Okay, and in that process, again, we were able to make this reduced coenzyme, which is going to be important for the electron transport chain. Okay, so now at fumarate, we're at this alkene. So what we see is water reacting with fumarate. And this is actually a reaction we've seen in the organic chemistry portion of this course, where when you have an alkene or a double bonded carbon, you can add water across that double bond, meaning you're going to add an OH to one carbon and then an H to the other one, while breaking that carbon-carbon double bond. So that is how we end up at malate. So now from malate, we can look in this gray region and see that what is happening is that this hydroxyl group that used to be here has now been converted to a carbonyl group. So this is a classic oxidation reaction. And we can see this is the case because NAD+, which is an oxidizing agent, comes in and is converted to NADH. So in this last step, we are producing another reduced coenzyme molecule. And we are generating our starting material, oxaloacetate, for the next turn in the citric acid cycle. So at this point, once we have made oxaloacetate, then we would need a new acetyl-CoA molecule to come in to start turn number 2 of the citric acid cycle. So as you go through these turns, you are going to be generating those important compounds, which were our reduced coenzymes, NADH, FADH2, GTP, and then these carbon dioxide molecules. Okay, so now let's try to use this analysis, meaning our ability to be able to look at these compounds and extract information and apply it to filling out this cyclic metabolic pathway. So this is our citric acid cycle, and what we are being asked to do here is to fill in all of the empty boxes. Now, yes, you can just memorize the citric acid cycle if you wanted to, but what I want to do here is to use our ability to deduce what is going on by just inspecting our molecules to be able to determine what is going to go into these boxes. And this is why we have spent so much time looking at organic chemistry reactions and then talking about the reactions of lipids, carbohydrates, and protein, in order to be able to look at something that is complex, such as this metabolic pathway, and be able to deduce information from it. All right, so how can we start this? Well, there are many ways that you can approach this. I'm going to show you the way that I would approach this. So first, I would want to identify where my acetyl-CoA molecule is coming in. So remember that your acetyl-CoA molecule is going to be these 2 carbons that are coming in. It's going to join these 4 carbons. So wherever I see, to make the 6 carbon compound, so wherever I see a conversion from 4 carbons to 6 carbons, that is the only time that an acetyl-CoA molecule is going to come into your cycle. So I can inspect all of my molecules along my cycle here, and then I recognize that the point at which I have 4 carbons here, and then I go to 6 carbons here, then this is going to be the point where my acetyl-CoA comes in. So if I count 1, 2, 3, 4 carbons, here I have 1, 2, 3, 4, 5, and 6. Okay, so that means that I have acetyl-CoA coming in as a substrate here. And this makes sense because I have coenzyme A being released, meaning that thioester bond has been cleaved, right? So this means I have acetyl-CoA reacting here, okay? All right, so that CoA is released, and then that acetyl group is added to that 4 carbon compound in order to make this species here. All right, so now I'm here. I'm going to continue along this cycle. So I know this was step number 1 here. So I'm going to continue, and then I will inspect this reactant here and the product. All right, so the first thing I notice is that this COO minus group, which is a carboxyl group, that is no longer present in my product. So what this means is that this group was removed as a CO2. And then in the process, we learned in the previous slide that in this reaction, when you're releasing CO2, we can also go ahead and reduce NAD plus into NADH. And to be complete, I will include the proton that is also produced. Okay. But the main point of this step is that we have produced NADH. Okay, great. So now let's look at the next step. In the next step, I see that I have coenzyme A coming in. And as I look at my molecules here, I see that this group down here, the COO minus, has been replaced with coenzyme A. So that means that I have lost this group. So that is a CO2 group. So I will release that as carbon dioxide. My coenzyme A will come in and take its place. And then simultaneously in this step, I will also produce my reduced coenzyme. So NADH plus H plus. All right, we're almost there. All right, so let's inspect the next step. So immediately I see that I have released coenzyme A. So this region of the molecule that was here is no longer there. Now we see it's been cleaved off, and now I have this the remaining 4 carbon compound here. So because I recognize that I have cleaved this high energy bond here, I know that I'm probably providing a lot of energy to another reaction that requires it. And in this case, I see I have GDP plus inorganic phosphate. Well, that means that I have phosphorylated GDP to GTP. So again, the energy that came from breaking this bond here is going into phosphorylating GDP. Okay, so I have successfully produced GTP. All right, let's do this last step. I see here that I'm going from this compound to the alkene. So what this means is that I have oxidized this. And I see that I have FAD as one of my reactants. So that means that I will be producing FADH2, because FAD was able to pick up those 2 hydrogens that I needed to remove in order to make this double bonded structure. So FAD will pick those 2 hydrogens up to produce FADH2. All right. So in the next step, we have water. This is a reaction that we recognize because this double bond is broken, and then you add OH and H to each carbon. And then lastly here, we see that we have this hydroxyl group here has been oxidized into this carbonyl group, right? Well, since it's oxidation, this means that I must have had NAD plus come in and pick up the hydrogen and release one of those protons. So I know it's NAD plus because we discussed that in this cycle, we only produce one FADH2. Okay, so that FADH2 was produced in the step where we produce this alkene. That is the only time we use FADH2. For all of the other oxidation steps, we are going to use NAD+. And so that is an example as to how you can fill in the gaps in a cycle, be able to deduce from just looking at the reactants and the products of a reaction, be able to deduce what is going on. All right, so let's summarize everything we talked about. So we can think about the citric acid cycle as one step. So if we just think about what are all the reactants that go into this cycle and what is everything that comes out, well, you're going to see that we have acetyl-CoA that is going to come in as a reactant. We're going to have some water molecules coming in as well. But I'm going to underline the ones that we are mostly interested in, the important ones. We're going to see that we have GDP and inorganic phosphate also coming in. That is what's going to produce GTP. And then we had our oxidized forms of our coenzymes, and then those get reduced. So this is going to be the output here. And specifically, it is 3 NAD pluses that are being reduced to produce 3 NADHs. 1 FAD molecule will be reduced to FADH2. The other important point here is that we're going to produce 2 carbon dioxide molecules, 1 in each step here. Okay. And then yes, we're also going to release some coenzyme A. So we can summarize this as follows. So we can think of one turn in the citric acid cycle, meaning when we feed one acetyl-CoA molecule and do just one turn along these 8 reactions, one turn is going to produce 2 carbon dioxide molecules, 3 NADH molecules, 1 FADH molecule, and 1 GTP molecule. So it's going to be these that you're going to need to know in order to be able to do problems like the ones we're going to do next. Again, I just want to reiterate here that the reason we're looking into this is because these reduced coenzymes, along with this GTP, are going to be important in calculating how much energy we can produce, because these reduced coenzymes are going to go into your electron transport chain, right? We will talk more about how those coenzymes feed into the electron transport chain to generate ATP in the next video. Okay, so let's apply these concepts of how much NADH we are producing. What kind of problems can we solve with this? Okay, so here's an example of one. Mystery nutrient molecule X gets digested and the products of its digestion enter the citric acid cycle, where they ultimately cause the production of 6 molecules of CO2. So what it's saying here is that this molecule X, it could be a monosaccharide, it could be a fatty acid, it could be an amino acid. Okay, it has been converted to this acetyl-CoA, and it has entered the citric acid cycle. Now we have produced a total of 6 molecules of carbon dioxide. So what this means is, let's just reiterate how much of each we produce in one turn. Okay, so in one turn of the citric acid cycle, actually let's organize it in this way. I'm going to write the molecule name here, and then how many I produce in one turn. So this is just summarizing the information on the previous slide. So CO2 is one of the compounds we're interested in. In one turn, we produce 2 carbon dioxides. For FADH2, we produce 1 of those molecules. And then for NADH, we produce 3, right? This is in one turn. But here they are telling us we produced 6 molecules. So this means we must have done more than one turn, because in one turn we only produced 2 CO2s. So what this means is that if I produced 6 molecules of CO2, that means 2 times 3 is 6, so we must have done 3 turns. Okay, each turn producing 2 carbon dioxide molecules. So since we did 3 turns, that means we are also producing 3 total FADH2s, and we're producing 3 times 3, 9 total NADH molecules. So to answer this question, that is all we had to do. So FADH2, we produced 3 because we did 3 turns. NADH, we produced 9 because we did 3 turns. And CoQ10, this coenzyme is going to be part of the electron transport chain. So since we're talking about the citric acid cycle, that is going to be 0, because no matter how many turns you do of the citric acid cycle, you will never produce CoQ10. All right, let's look at another example of how we can use that information. Suppose the following nutrient molecule is digested and the products of digestion enter the citric acid cycle. It is below the important products that will be formed out of this molecule by the citric acid cycle. So again, we're interested in looking at NADH, FADH2, GTP, and CO2. Okay, so here they have given us a fatty acid. So you'll recognize that you have your carboxylic acid head here. So here's your carboxylic acid group, and then this long carbon hydrogen tail, which is classic of a fatty acid. So the key to solving a problem like this is recognizing that the acetyl-CoA that is coming in to your citric acid cycle to generate one turn of the citric acid cycle, those two carbons are coming from your building blocks. So what that means is that these two carbons here are going to be what comes in as two carbons in acetyl-CoA. These are going to be bound to acetyl-CoA, or they're going to be part of acetyl-CoA. So those two carbons are going to come in, and it's going to generate one turn. So those two carbons represent one turn of my citric acid cycle. That means that then these next two carbons are going to represent turn number 2. Then this represents turn 3, turn 4, turn 5, 6, 7, and 8. So here I am undergoing 8 turns. Another way to do this is you can just count the number of carbons that you have, which is going to be 16 carbons in this case. Then divide that by 2 because you have to put 2 carbons per turn. So 2 per turn. So that means you get a total of 8 turns of your citric acid cycle. So from this point, it is pretty easy then to just calculate how much of each molecule you would produce. So let's start with CO2. So in one turn of the cycle, I produce 2 CO2s. So if I'm doing 8 turns, that means 8 times 2, I will produce 16 CO2 molecules. For NADH, I produce 3 NADH per turn. So since I have 8, 3 times 8, that's going to give me 24 molecules of NADH. Now let's look at FADH2. Well, we only produce 1 per every turn. So since we did 8 turns, we're going to produce 8 of these. Then lastly, we're going to look at GTP. We produce just 1 GTP per turn, so that will be a total of 8 GTP. So this is how understanding how the reactions in the citric acid cycle are occurring and what the important products are and how many of each you are producing. This will inform you into being able to calculate how much of each molecule you can produce given a particular building block. Okay? So in the next video, we're going to talk about what happens to these NADH molecules when they are fed into the electron transport chain and how it is that those reactions that are going to follow will generate more ATP molecules in the electron transport chain.